Yesterday we had discussed about the structure of protein kinase, and the structure of protein kinase basically now you understood. This is having two lobe structures, smaller N-terminal lobe and larger C-terminal lobe, and it has got different structures like this is all in the activation loop. So P loop is there, C helix, catalytic loop, activation segment with the activation loop, then autoinhibitory elements. So these structures of protein kinase are responsible for the functional part of this protein kinase. So now the inactive and active mainly difference is the closed and open structure and then phosphorylation. Okay, so the active site is having phosphorylated residues. And if you recollect yesterday we discussed autoinhibitory element is different from activation loop. So it is not activated because of the phosphorylation like in case of activation loop, rather it is influencing the activation pouch to get closed. So that is what the way how it functions, autoinhibitory element, and that is why it is known as pseudo substrate, right? So I hope all of you can recollect very well because yesterday only we discussed about all these structures. P loop or glycine loop, C helix is the important for regulation of this protein kinase. Then catalytic loop we discussed, and then activation segment with the activation loop. So here is the important because you have the tripeptide motif is there, and there are several times it is repeated, and this represents the activation site where the substrate peptide is going to bind, and then the P loop is the region where ATP is bound, and from that it is responsible for transfer of phosphoryl gamma phosphate group to the peptide. And apart from these, which have got the role in the function of protein kinase, there is autoinhibitory element which is responsible for negative regulation in case of protein kinases. Now there is one question for you. Today you are writing exam. What is the negative regulation in case of G protein coupled receptors? Which is responsible for negative regulation? Somebody should tell fast. This is very easy if you are very well prepared for the exam. So negative regulation of G protein coupled receptors.
Gap proteins. Gap proteins.
Okay.
RGS protein
Very good.
and phosphodiosin, sir.
Okay. Then another important thing I am expecting that you are not telling. RGS protein, very good. So then another one, very important. You people didn't prepare. I given important questions also. I am happy because you people are able to tell which is negative regulation. That's good. But one important thing I am expecting from you. That is one of the subtopic in your syllabus. Please tell, Sanjana. Like the heterotrimeric G protein coupled receptors are having another effector molecule which is responsible for negative regulation. Which one is that? You all know about it. Tanisha, Vidya.
Sir, is it not gap GTPases?
No, as far as negative regulation is concerned, you are correct. Gap proteins, okay. But the immediate effectors of G protein coupled receptors, like heterotrimeric gap proteins, where it will come, that is G domain and then phosphorylation. But I am telling G protein couple GPCR transmembrane receptor, the activators are G proteins, right? Heterotrimer G proteins are attached to the cytosolic end of the G protein coupled receptors. Like that, there are other proteins or effectors which are responsible for negative regulation of the GPCRs. What are those? So now I am unhappy because that is very important question. I think I given in your today's question, and also please note I written your questions like Roman 1 and Roman 2, two sections. This is like earlier our internal exam pattern. So Roman 1 you will have 6 questions, 5 questions you can answer, and Roman 2 we can answer question 4 questions, 3 questions you are expected to answer. Okay. Okay, tell me, Pooja, like RGS, you should tell one more thing. That is the immediate effector of G protein coupled receptor. Last topic. I given the question in your important questions also. Have you not read arrestins, Tanisha? Arrestins, you people have not prepared. That is very important. Okay.
Yes, sir. Yes, sir.
So arrestins are responsible for negative regulation. Okay. So that is what the negative regulation in case of G protein coupled receptors. Whereas in case of protein kinases, there are autoinhibitory elements apart from the activation group which will negatively regulate. So protein kinase reaction, what is that? What is the function of protein kinase now? Nothing but phosphorylation of the substrate protein. And in signal transduction, that substrate protein on which protein kinase is acting is another protein kinase itself, right? So now we discuss the classification also, you are well aware, different type of protein kinases. So serine, threonine, and tyrosine, specific protein kinases, we discussed already that they have common catalytic function of covalent phosphorylation. So this is a covalent attachment and it's a reversible process. So covalent phosphorylation of the substrate protein via transfer of gamma phosphate of ATP to the hydroxyl group of serine, threonine or tyrosine residue. So this was the first sort out function of protein kinases. So serine, threonine and tyrosine were the first protein kinases whose functions and structure was well elucidated. And in that case, the protein kinase reaction means covalent phosphorylation of the substrate protein via transfer of gamma phosphate of ATP to the, these are all hydroxyl group containing amino acids, so hydroxyl group of serine, threonine or tyrosine. So this is the common function. This all you know about it. So basically kinases are phosphorylating the substrates and reversing because of the phosphatases. So now this protein kinase catalytic function is carried out by the catalytic domain of 270 amino acids which are highly conserved. So the amino acids in the catalytic domain motif of the protein kinase are highly conserved. Means in all the protein kinases these amino acids are invariable. They are the same amino acids that will be present in the catalytic domain. Those are above 270 amino acids. Now the key players of this protein kinase reaction, that phosphorylation. So that is amino acids which are acidic in nature. Acidic amino acids and basic amino acids present in the catalytic domain have got very important role. So this is protein kinase reaction. And firstly, acidic amino acid, what its role? Stabilization of the transition state. What is transition state here? Open and closed. So from closed structure, it forms the open structure. So that stabilization is because the acidic amino acids. And then the second one is activation of the hydroxyl group of the acceptor amino acid for nucleophilic attack on the gamma phosphate. So hydroxyl group where it is on the substrate. So substrate hydroxyl groups to which phosphorylation has to take place in the substrate protein kinase has to become active. So this active hydroxyl groups, they will form a nucleophilic attack over the the gamma phosphate group of the ATP. So next one or two metal ions. This also you can underline. So first is acidic amino acids. Then second one is metal ions. One or two metal ions. What is its role? They coordinate the gamma phosphate of ATP. Yesterday we discussed, right? So this ATP in its structure is getting stabilized in the smaller lobe of the protein kinase, and in that, these metal ions are responsible for stabilization, holding the ATP within the protein kinase. Okay, so this help to fix the ATP and stabilize the negative charges in the transition state. Okay, so the negative charges in the transition state are stabilized. Like in RNA polymerase structure, all the time we were discussing this transcription bubble formation, and mainly all these subunits will function in such a way that the elongation complex or pre-initiation complex will be stable structure, so that the conformation will be stabilized in such a way that the DNA is unfolded, and then it forms transcription bubble so that it provides the sites for synthesis of RNA. So the same way here, stabilization of the protein kinase structure because of the conformational change from the closed conformation to open conformation. Closed is inactive, open is active. In that, there are stabilization factors which are responsible for the stable transition state to go on. So basic amino acids, acidic amino acids, these two functions, stabilize the transition state and then activate the hydroxyl group of the acceptor amino acid. Then basic amino acids, apart from metal ions, these also functions in stabilizing the negative charges in the transition state and in the leaving group of ADP. So negative charges in the leaving group, that is from ATP, and then during the transition state, they are mainly stabilized. So all these stabilization factors responsible for what is a very important attribute or parameter associated with the activity of proteins, I said. Can anybody tell? For protein activity, which is the parameter? What is important or what is taking place here? Can anybody tell? Please speak out. Let it be wrong. No worries. Alodi,
Phosphorylation, sir.
phosphorylation is the step which is responsible for what happens if phosphorylation happens. What happens to the protein? In signal transduction, phosphorylation is the trigger. But if phosphorylation is happening, what is happening then? So that it is altering its activity, functionality of the protein. You have to think, okay? So when you are studying, you have to think. That is missing. Tell me, Pooja,